Hello boys, welcome back to another session of online biology video tutorials, and this session is meant for class 12th biology students, and this is meant for 30th June 2021, as we have been discussing about chapter number 2, sexual reproduction in flowering plants. And in yesterday's session we talked about the structure of the flower in general, and then we talked about the four ovules that will be there in any flower, right from what we call calyx, corolla, androecium, and gynoecium. And we said that androecium is the male reproductive organ, particular male reproductive system, and that system has got male reproductive organ called stamen. And we discussed about the how anther will be present and how the microsporogenesis will take place, in what way PMC will be able to form microspores, and the structure of a microspore and the role of sporopollenin. And we also talked about the formation of pollen grain, and pollen grain having vegetative cell as well as generative cell. And we also said that the generative cell is small, spindle shaped with a dense cytoplasm and having a nucleus, and this nucleus undergoes mitotic division, forms two nuclei, male nucleus 1, male nucleus 2. So with that background keeping in our mind, we move on to the fourth ovule, most important one, that is female reproductive organ, and the system is called gynoecium. The ovule is called gynoecium, and the gynoecium will have female reproductive organ, namely pistil. So pistil is the female reproductive organ, and I said the female reproductive organ is called gonad, and the gonad undergoes meiosis and forms gamete. So female gonad is pistil or carpel in a flower, and the gamete is ovum, right? So keeping that in our mind, the gynoecium, the fourth ovule, consists of stigma, style, and ovary, and of course below the ovary there will be a thalamus structure. And you have got this hibiscus pistil, which you must have seen. Even if you have not seen, if you got a hibiscus flower, hibiscus plant in your house, please check it today itself. You just remove the petals, remove the sepals, remove the anther, and you can see the structure with your own eyes. Then of course multicarpellary syncorpus pistil in papaver. This way you can see some of the flowers having this. Then multicarpellary, many carpels, apocarpus, gynoecium means separate carpels in mycelia. So basically gynoecium is a female reproductive part. It may have a single pistil like this, okay, or more than one pistil. If the flower has got single pistil, we say monocarpellary, one carpel, mono. If the flower has got more than one pistil, then we say multi, means many carpels. So in multicarpellary, the pistils may be fused. If they are fused like this, we can say syncarpus, and if they are free, then we say apocarpus, like in mycelia. Moving ahead, basically the structure of the flower we have been talking about. As I said, the group of sepals, green colored, calyx, group of colorful and attractive with a lot of pungent smell, their petals, corolla. Then you got androecium with the stamens, and you got a stigma, style, ovary, which makes carpel or female gynoecium. So each pistil has basically three parts. The stigma part, this one, okay, style, elongated, slender part beneath the stigma, this portion, style, and then you got ovary, basal, bulged portion, this one. This is how a pistil has got three parts. So a pistil has a terminal receptive disc-like stigma, a stalk-like style, and a basal swollen ovule bearing part called ovary. Inside the ovary is the ovarian cavity with one or more locules and parenchymatous cushion called placenta, from which the ovules arise. So basically this is how you remember the gynecium part of it. And then further we moving ahead, inside the ovary, as I said just now, ovarian cavity, locule will be there. You can see here the presence of locule, ovarian cavity, and arising from the placenta are the ovules. Now the ovules are called megasporangia. The number of ovules in a ovary may be 1, like in wheat, paddy, mango, etc. In some like papaya, watermelon, orchids, there will be many ovules, multi-ovules. So basically you got this stigma, style, and the bulged portion is ovary. Inside the ovary you got that locule room, ovarian wall, and then there is a locule room, and inside the room you got ovules. So this is how the presence of ovules will be there. And the structure of megasporangium, that is ovule. Ovule is attached to placenta by a stalk called funicle. Now look at this. We are expanding this portion, ovule portion. So this is ovule. Yes, right. This ovule portion we are enlarging here. Right. So ovule is attached to the placenta by a stalk called funicle. Look at this. This is funicle. Right. And junction between the body of the ovule and the funicle is called hilum. This is the junction, hilum. The junction between the body of the ovule is this, and the funicle is hilum. Each ovule has 1 or 2 protective envelopes. They are called integuments. So you can see here presence of outer integument and inner integument. 2 integuments are there. And then there is a small opening at the tip, and that small opening at the tip is, you must remember this, micropyle. Micropyle always is present on the top. Opposite to the micropolar region or end is the Cellulase. Look at this. There are 2 poles, north pole, south pole. Micropolar region or micropolar pole or Cellulase pole. So in the center, in between, you have got the highly nutritious tissue called nucellus, and then embryo sac. Embryo sac will be there. So further you can see the locules particularly. So this is the placenta, this is the funiculus, these are the ovules, because ovules are the megasporangium, and this is the room, locule, and this is ovarian wall. So this is how you must understand that megasporangium, that is ovule, will be situated inside the gynecium. And further, enclosed within the integuments, see, outer integument and inner integument, there is a mass of cells called nucellus. This is the nucellus, this region, nucellus, mass of cells will be there. And its cells contain reserve food materials. That is how they are nutritive, highly nutritive tissue. This helps the growing embryo. Inside the nucellus is the embryo sac. This is actually female gametophyte. This is the embryo sac, this one. And an ovule generally has a single embryo sac formed from a megaspore. So the megaspore forms an embryo sac. And remember, there is a north pole and south pole, micropolar region, and Cellulase. So this is how a ovule, megasporangium structure will be present. And how the megasporangium forms megaspores. That process of formation of megaspores from megasporangium is Megasporogenesis. Genesis means taking birth. What is taking birth? Megaspore. From where it is taking birth? From MMC, megaspore mother cell. But one precaution to you, yesterday also I used this MMC. There I used microspore mother cell. But here also I am using MMC, but I am saying megaspore mother cell. So to avoid that confusion, what you say, you use better PMC, pollen mother cell for microspore. Whereas for megaspore mother cell, you can use MMC for formation of megaspores. So it is the formation of megaspores from megaspore mother cell, that is megasporogenesis. So ovules generally differentiate a single MMC in the micropylar region of the nucleus. See, this is the micropylar end, and these are the nucellus tissue, nutritive tissue, and you can see the megaspore mother cell. This is the megaspore mother cell. So it is this cell which undergoes meiosis and form megaspores. So where is it located? It is located here in the embryo sac. And further, when this megaspore mother cell starts dividing, you can see formation of dyad, megaspore dyad. Two cells are formed, 1 and 2. And then tetrad, 1, 2, 3, 4. Megaspore tetrads are formed. So MMC basically undergoes meiosis to produce 4 megaspores, just like the pollen mother cell undergoes meiosis to produce 4 microspores. They are microspores, here megaspores. And then in a majority of flowering plants, 1 megaspore is functional while the other 3 degenerate and become food for the functional megaspore. So we use the word functional megaspore. So the functional megaspore develops into female gametophyte. So female gametophyte is formed because of the megaspore. Now look at this. This is the megaspore tetrad. Now this is the chelagel end. Integuments are there. Meiotic tetrads are formed. Now out of the 4, look at this. Only 1 is surviving. The one that survives is the functional megaspore. And other 3 get degenerated and become food for this functional megaspore. So this is how the functional megaspore is formed. So the embryo sac formation from a single megaspore is called monosporic development. Since there is only one functional megaspore, it's a monosporic development. And then the nucleus of functional megaspore now starts dividing. So this is how the formation of female gametophyte, how embryo sac is formed. Now we understand with the different stages. The nucleus of the functional megaspore divides mitotically. Now it is mitosis to form 2 nuclei. Now look at this, 1, 2 nuclei, forming 2 nucleate embryo sac. The nuclei again divide 2 times, forming 4 nucleate and 8 nucleate stages of the embryo sac. Now look at this. This one forms 2 nuclei. This one forms 2 nuclei, 4. And again these undergo mitosis, 4 plus 4, 8. So 8 nuclei are formed. So you can see 2 nucleate stage, 4 nucleate stage, 8 nucleate stages of the embryo sac, and a mature embryo sac is formed. And these divisions are free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. Only nucleus is formed. Now you can see out of the 8, 3 will be coming down, they are called antipodals. 2 will be in the center of the embryo sac, they are called polar nuclei, it is central cell. And another 3 will be, out of the 3, 2 are called synergates, and 1 is called the egg cell. This is important for us. This is the functional one. That is what we say female gamete, egg. So out of the 8 nucleate, 3 down, 3 up. So 3 plus 3, 6. Centrally 2 polar nuclei. So 6 plus 2, 8. This is 8 nucleate embryo sac, right? Now, out of the 8 nucleate sac, now only until now nucleus is divided. Now cytoplasmic walls will be formed and cells will start forming. So formation of female gametophyte embryo sac. After the 8 nucleate stage, now this is important, cell walls are laid down, leading to the organization of typical female gametophyte. This is how the female gametophyte is formed. 6 of the 8 nuclei are surrounded by cell walls. Okay, 6 you can see the 3 antipodals and 3 synergids. They are all surrounded by cell walls and organized into cells. That means 3 cells top, 3 antipodals, 3 synergids. So 6 cells are formed out of the 8 nuclei. And the remaining 2 nuclei, that is the nuclei which are there, polar nuclei which is in the central cell, they are situated below the egg apparatus. Okay, below the egg apparatus. This is that, and it is a large central cell. Now these 2 nuclei will be having their own wall like this. So how many nuclei are there? 3 top, 3 bottom, 6 plus 2 centrally. So 8 nuclei. 8 nuclei is there. Okay, that's clear now. 8 nuclei. How many cells are there? Here 3 cells, here 3 cells, 6, and here 1 cell. Because both the polar nuclei are forming only 1 cell. So 3 plus 1, 7 celled. So we say this embryo sac structure as matured one having 8 nucleate, 7 celled embryo sac. Okay, that is the matured one. Now, distribution of cells in the embryo sac. A typical mature embryo sac is 8 nucleate and 7 celled. 3 cells are grouped at the micropolar region and form egg apparatus. You can see micropolar region is this, and you have got 2 synergids and 1 egg cell. So these are the 2 synergids and this is the egg cell. Now this is the egg cell. This is most important. And these are this one and this one. 1 and 2 synergids, 1 egg cell, and central polar nuclei, and then 3 antipodals, 1, 2, 3. And so 3 cells are grouped at the micropolar region and form egg apparatus. This whole thing, synergids plus egg, is called egg apparatus, right? This is egg apparatus. And synergids have special cellular thickenings at the micropolar tip and called filiform apparatus. This one, this finger-like structures, filiform apparatus. It helps to guide the pollen tube into synergid. This is the function of filiform. 3 cells at the cellulase end are called antipodals. C cellulase antipodals. Ca, you must remember. Cellulase end antipodals. Micropolar region synergids. MS. Microsoft. Okay. And the large central cell has 2 polar nuclei. This polar nuclei will be there. Now, structure of the flower, formation of female gametophyte overall. So this is the meiotic tetrad, degenerating megaspores. 1 functional spore will be formed, and in the stage 2 this nucleus becomes mitotically 2. These 2 will become 4 nucleate, 4 nucleate will become 8 nucleate, 8 nucleate will be arranged in this form. So mature female gametophyte will be having a structure like this. So you got cellulase end at with antipodals Ca, and you got synergids with egg apparatus. So this is how the female gametophyte matured embryo sac will be formed. So keep on drawing the diagrams in your notebook, and then you can understand how this matured embryo sac will be formed.